Give me the thumbs up, David.
Yeah, I'm ready. Are you ready?
Okay. All right. Well, welcome aboard, everybody. So we are here. We are part of the Stratovan Corporation. We're a small company based out of Northern California. We specialize in software development, and as you can probably tell with the segmentation software suite, we focus primarily on the development of detection algorithms. And so part of that development process is, of course, being able to work with 3D data. And this tool set was developed in-house, architected by Dr. David Wiley, who's sharing a screen, and presently the other person, on the call here, getting ready to present. And he and the team here were able to develop a suite of tools to be able to work with 3D data, find the objects that they're interested in, and segment them in a way that now an AI or a machine learning based algorithm can ingest that data, make it a high quality, make it easy to do, and now you get better performance out of the algorithm when your data is a little bit more pristine. So Dr. Wiley has been working on this since at least 2005. The company Stratovan was founded in 2005. David's got his PhD in this field, has been working in this for far longer than that. And as a company, we have an extensive history working with TSA and the medical industry in both object detection within the human body to find abnormalities or to help with surgical planning, and then on the TSA and Department of Homeland Security, looking for objects of interest in things like cargo or luggage. There's a lot of uses here. I think I'd speak a lot to not just the detection element, but if you are looking for publications and you want nice, clean segmentations of objects of interest, this is very useful for that. This is useful in the manufacturing world. This is useful in a lot of different industries right now. So without further ado, I'm going to turn it over to Dr. Wiley. He's got a couple specimens and a couple scans that he's going to use to demonstrate the tool sets. He'll walk through it. We would like to invite anybody to ask questions, and we can answer them as we go. But go ahead, Dr. Wiley. Thank you.
Yeah, thanks, Ryan. So what I'm going to do is I'm going to start here by just loading a brain scan. It's actually mine, so it has my name there. So this is our software called ProSurgical. And you can point to, different file types here. So in this case, I want to load the DICOM file from this folder, which I just did here. And then this tool provides some basic 3D visualization, the slice windows, which you can see over here. And you can manipulate the 3D data here. For the purposes of this webinar, you can look at this mouse down here in the lower right-hand corner, and you can see the buttons that I'm clicking. So here I'm left clicking, right clicking, mouse wheel, middle mouse click. If I hold down the keys to do different operations, you can see those control and shift. That becomes useful later on when doing segmentation and that kind of thing, so you can see how it's actually being done. Just to give you a quick overview, so in this view, you've got the volume rendering that's going on here. Then you have the slices. If you double click in the windows, you can swap them around. If you have the big window here, you can have it fill the whole screen. And in this case here, I'm doing a mouse wheel to scroll through. You can also grab this, scroll through here. On the surface side, on this over here, we have a histogram. You can move this line up and down to pull out different kinds of surfaces. This stuff up here is an artifact of this particular MRI scan. So you can get some surfaces here. This is more interesting with the CT scan, which I'll show in a bit. Do some cropping if you want to look inside of the different objects. You can see that. If you have two scans of the similar things, you can load two side by side. And now what I'm going to do is jump into the segmentation capability and just show you a couple things on this particular scan. Ryan, if there's any questions or anything, just make sure to interrupt me because I can't always read what's going
No,
on.
yeah, you focus on that. I'll let you know if anybody has any questions or wants to see anything specific.
Okay, so on the segmentation tab here, there's 3 different ways that you can create and manipulate segmentations, which you can start up with in this box up here. So there's a manual segmentations. We actually don't recommend creating them manually. The manual tools are basically for editing. There's a semi-automatic segmentation capability and a thresholding. And what I'll do is start by demonstrating some of the thresholding capability. So you can see with the cropping, if I use the crop lines, that can limit the area in which I do, which the segmentation algorithms are applied. So in this case, if I want to try and let's say segment the ventricles here, ventricles are this dark area. I can move, this is the histogram which represents the data values that you see over here. Bring in the crop region. Focus on this area here. And when I bring in that crop region, it updates the histogram on this side over here. So the purple area has been automatically segmented based on this range, this blue range here. If I want the ventricles, which is the dark range, I need to move this down, and this will exclude the higher intensity tissue regions and focusing on the lower intensity regions. So in this case, we pulled out ventricles there. This is some part here. So the challenge of segmentation, however, is how do you separate, the object of interest from things that you don't need? And a lot of times, thresholding can work well, or doing the segmentations just based on the thresholding can work well, but in this case, you can see it bleeds all over the place. So in this particular example, this capability is not ideal. And a better option is actually to switch over to the automatic segmentation. And what I'm going to do is I'm going to clear this. And the idea here is I'm holding down the shift key here. You can change little box size. And what I'm going to do is I'm going to say, I'm left clicking. I'm going to say, I want these areas. I don't want these areas. And if you do that in a couple parts of the object that you're interested in, I want that, I don't want that. And then hit start. What that will do is it'll start to pull out that object in those areas that you selected. So here I've traced it down a little bit, and I want to go to over here. You want to try to balance the number of red and green dots that you place, and then you can kind of continue to expand your selection. And I'm ignoring the little bits and pieces that are missing right now, and I'll show you how to correct those. But my goal right now is I want to separate the left and the right ventricles, which I have 2 different colors here, and that tells me that I did separate those. Say I wanted to expand up into those little crevices. Expanded a little bit too much, so I'm going to put, if I don't want these to touch, I can put some blocking dots in there. also say that I don't want this stuff out here. Okay, so let's take a look at this. So you might be looking at this and thinking, well, there's little holes in there, and I'll show you how to deal with those in a second. But I'm looking at the red and the blue as I'm going through, see if I'm happy with those. Just initially here. Maybe I want a little bit more here. Let's restart it. Okay, let's stop there for a second. Now what I want to do is I have all these objects that are pulled out over here. And this is a complete list of all the different objects. And as I click on these, it moves the crosshairs here around. I can also, from one of these windows, I can hold down the Alt key and then left click, and that will select the object. So in this case, it's selected object number 1. And if we look here, object number 1 is the blue, and object number 2 is the orange one. And those represent ventricles. So what I'll do is I'll now move these over, and this is these are like temporary objects that you use for doing the process of segmentation, and then when you want to make them permanent, you move them over with these little buttons over here. And then now what we can do with our left and right ventricles, we can worry about filling in with these little holes that are here. And so best way to do that, and I'll find a good slice here. Best way to do that is to use something called the morphologic operations. And so when I have an object selected from here, I can, from this list down here, choose different operations that I can apply to that object. So in this case, I'm going to do something called dilation. So dilation is going to expand the segmentation. And so I've selected that. And then if I hit apply here, you can watch what happens to the green segmentation there. And it grew and it got bigger. And it filled in the little holes. And then you can do the opposite, which is called erosion, and then erode. And then that will actually shrink it back down. And so we expanded the segmentation and then shrunk the segmentation back down. And what that does is fill the little holes that you have. So I'll do that again with this one on the right-hand side. The purple one, which I've selected, and that operation, the dilation and the erosion, is actually called a closing operation. So it's the combination of those two. So we have presets here. And if I choose this one, this will dilate once and then erode once. If we do these down here, it'll do 2 dilations and then 2 erosions. And the opposite, if you want to actually make holes bigger, you can do something called an opening. But in this particular case, we don't want to do that since we want to close these little holes that are there. So I'm just going to click this button here, and that's now filled in all the little holes there. Let's say we want to add this part down here. Let's say I'm not happy with this, and I want to add, this little leg down here and this other one on this side. I can go back to my segmentation capability. In this case, I'm going to use the threshold to try to grab these little areas here. So it's pretty close there. Bring this down. So I used the automatic segmentation to get, a large part of it. And now I can use this threshold segmentation, and you can see the low density peak right here, which likely represents the dark region of the ventricles there, or the cerebral spinal fluid. And then you can manipulate the segmentation threshold to isolate that particular part. And so in this case, you can see I've got this orange ventricle here. These are the ones that we segmented before. Now we've got these little pieces here. And let's see. All right, let me see if I can select that again. Okay, so those are the two little pieces that I want. So it's that one there. So I'm going to move that piece over. And then that piece there, move that piece over. And then I can go back to the manual segmentation capability. Now I've got all four of these pieces here. And what I want to do is now join these little bits and pieces to that little leg that I just made there. So the way to do that is I've now selected these, holding down the control key to select multiple pieces. You can see this is now joined or connect. Well, it's not connected yet, but it's those two pieces, the green and blue, are selected. And then I can go over here and I can click on merge. And what that's going to do is that's going to merge those two pieces together. And I click on that into a single object. So that's now a single object there. And then I can do the same with the other side. Merge those. If we want, we can do some of the closing operations to fill in the holes and clean it up a little bit. That's pretty much the process there. And so this ends up being a pretty good example to show partly how to use the automated tool in conjunction with the thresholding tool to build objects that you need. From the manual tools perspective, these allow fine-grained corrections to your segmentations. So here I've got the green object selected, and if I hold down Shift and then do left click, I can paint the segmentation on there. If you do Control Z and undo it, you can clean it up. But if I need to do some fine-grained corrections of the segmentation, I can get in there and do that for either object. So in this case, if I have the green object selected and I'm making edits to that object, I can't paint over the other objects. So you see that how the purple one there is protected. So that helps you maintain the integrity of your various objects that you're working on when you're doing that. I think that covers. Does anybody have any questions or anything on kind of that process of using the automated tools or the thresholding tools? Let's take a pause there and see if anybody has any questions.
No, I'm not seeing any yet.
Okay. Okay, so there's a lot of tools out there that do segmentation sort of along these lines. And one of the main benefits of this particular tool is we have different batch operations. So we go beyond just the basic segmentation capabilities, like I just showed you. So while we can do those, we also have capabilities for processing thousands of files at a time, reviewing quality assurance of your segmentations, and also tracking operator performance. So if you're going to do a batch of several thousand scans, and you've got, let's say, a group of 10 people that are going to be using segmentation software to be able to isolate objects like this, then label them and that kind of thing, it's useful to have performance metrics of the operators, for example, to know how much time and effort they've been spending on things. So I'm going to save this here. So I'm saving the work that I just did here, and I'm going to jump to just to quickly show you operator performance for this particular object. So if I put in the, oh, that's the curse of the live demo. Doesn't like this. Oh, give me a second there. I'll show the operator performance a little bit later. Okay, let's skip that part for now. So let's go to some of the batch operations. So I'll go over and show what these can do. So if you already have several segmentations and scans and that kind of thing that you've either generated with other software or some other capability, you can use this to import that data into the analysis tools that we have. So just very briefly, scan here. So I've got a folder of some data that I obtained from this challenge data set or website here. So these are CT scans with the aortic vessel tree segmented, and we're able to download this and end up getting, a bunch of different patient scans here, which are in different formats here. So this is the NRRD format, and you can actually convert. There are some built-in file conversion capabilities here. And for example, I can point to that folder, tell it what kind of file it is. So I could say it's an NRRD file, and I want to convert it to a different format. And then I can have it go through and do that conversion for me. So in this case, I could go here. They're selecting just 2 files. If I go up 1, it'll select all the files that are in there. And then, sometimes there's errors in the files. Spacing might not be set correctly or something like that. So you can actually correct some of that over here. So I just wanted to point that out. There's built-in file conversion capabilities. I've already converted them. And what I'm going to do is now switch to show how to do the importing. So in this folder here, I've converted them over to Nifty files. And then to do importing, what we want to do is create this proseg file here. So actually what I'll do is, okay, so this is the original. So I'm going to make Nifty files. Let's put them in this new folder here. Then hit convert, and it's done the conversion. You can see now it's made all the subfolders, and it's now converted them to Nifty files there. And then to do the importing process, go back to batch operations, import, select your file type. So in this particular structure that they have, the seg files are actually the segmentation files. So what I'm going to do, and they're in the same folder. So I'm going to put the same folder here. So I have my scans and images over here, and I have my segmentations over here. So over here, I want to filter and only keep the seg files. So I find, a common string that's in the file names, and I can put it there. And in this case, I want to exclude them. Oops. I want to exclude them. So now I've got my scans here, and I've got my segmentations here, and everything's in order. And then you can hit import all, and it's going to now load each of these scans, load each of the segmentation capability or segmentation files, and then it's going to create a new file that we need for the software, which is called the ProSeg file. So the ProSurgical segmentation file is what this is creating. So that's what's adding this. This file here includes the segmentation capability and additional information that we need to do various analyses. And then once this is done importing, you can now go into, you could export it. The other thing we can do is you can now load those files. So I'm going to go back here. Go to my folder here. So all of these are showing up. So I can select one of these scans to now load, and you can see that this is the scan. Not sure what this stuff is. This is the segmentation that was loaded in there, and then you can see the person that's in there as well. Currently, there's no labels here, and so if we have an understanding of what all the labels should be for the objects, we can also add that through the batch operations here. So we can go to let's keep this, edit segmentation labels, put that folder in there, gives me a list of all of my files, and then I can say, this tells me all the different object values that are in use. So the green aorta segmentation that you saw prior has a particular integer value that's assigned to it. And so what I can do is I can say, everything of value 1, whatever that object is, I can give it a certain anatomy. And in this case, it's the aorta vessel tree, for example. And then what that's going to do is assign all the objects of number 1 to that particular label. And it's done. So now if I go back to this object, I'll reload it, go back to this object, you can see it now has a label with ABT that's been assigned to it. This is important when you get to the quality assurance step. So from a quality assurance perspective, from all of these scans, it now tells me all of the different objects that have been identified within each of those scans. In this particular data set, there's only one object. It's not that interesting. So what I'm going to do is show you this other data set. So Tooth Fairy, this is also from Grand Challenge, and they have segmented, many different objects that are segmented in this data. So what we did is we actually did the same thing, downloaded the data, imported it into the ProSurgical file format, and then went through and added all of the labels for each of the objects. And in this case, labels, there's multiple labels here. And so you can look at a particular tooth or lower or upper jaw area, and you can see information like that. So what this list is doing is has a representation of every single object from every single scan. And so over here, you have histograms that are showing um, where each little box represents one of those objects in one of those scans. And the goal here is to um, find outliers to fix problems. Basically, if you've done a large scale segmentation effort, thousands of scans, dozens of people doing it, it's very easy to make mistakes. And if you have any mistakes go into a machine learning training pipeline, those mistakes translate into, poor algorithms basically. So the goal of this particular tab is to help identify and find problematic scans. So it's important to properly assign labels to each of your objects. So what I did earlier, not as part of this webinar, but I created a attribute database for this particular scan set. And so in this particular case, they provided, as part of the data set, they provided basically a JSON file that looks like this to represent the segmentation labels. And so, object 0 is background, that's nothing. Object 1 is the lower jawbone or the mandible. Number 2 is upper jawbone, and so on. So if we break this down into the different parts of the anatomy, and we have, so I create an anatomy label, which is the name here. It's going to be assigned per object, and it's categorical in nature. So I'm going to choose from a category of that anatomy. And then we can specify all of those different anatomic regions here. We can also specify things like, left and right side, upper and lower, first, second, third tooth, for example, central, lateral position. So we've created this attribute database here, which we can then apply. And so that has been applied to the segmentations. And then what the quality assurance view allows you to do is it's now looking for outliers within those groups. So right now it's not grouped very well. We have, premolar teeth that are all separated. And so what we can do to group things better, so just for example, we could put everything in the left group. Everything that's on the left side is in a group. Everything that's on the right side is in a group, just by clicking that over and giving it a second. So now what this means is all the objects that were marked as being on the left side are now grouped together and then displayed in the histograms here. And then all the objects on the right-hand side are displayed here. Everything here in the empty group represents things that don't or haven't been assigned the attribute left or right, if that makes sense. So in this case, we don't want to group, we don't want to actually use the left or right to group in this manner to find outliers, because you can see there's different kinds of things. But grouping based upon the anatomy is useful. So let's say I want to look at all the canine teeth together and all the incisors together. I can add the anatomy group here, and then that narrows down the groups that I have there. So all of the bridge work that's been done. This represents all the canine teeth, any crowns that were used in this particular case, any implant that any of the patients had, and so on. And then what you can do from these views is explore outliers within these different groups. And so, we might look at, so you see these histograms show different characteristics of the objects. So this top one is object volume. We have the mass in this one, and then the mean intensity value of the object there. And when hovering over an object, you can see the little yellow box in all three of those histograms is showing the same object. So if I look at this object here, this might be an outlier in volume, but it's not necessarily an outlier in the mean value, which was down here, for example. So you can see the yellow box is here, and then it's here, and then down here somewhere. So to find objects that you should probably take a look at, this list right here ranks the outliers in highest importance. So anything at the top of the list is likely an outlier. Anything at the bottom is likely not an outlier. So for example, if I pick any objects here, you can see that the little yellow box is pretty much always in the middle of the distributions. So this is in the this selection here is represented by this blue and now yellow selection here. And you can see that's right in the middle of the distribution. So things at the bottom of the list are pretty much central. Things at the top are outliers. And so if we click on the Fahrenheit up here, this outlier value of 10 shows that it is a big outlier. And then if we look at where it's falling, it's this blue box here that's been selected. So up here, argue in terms of volume, maybe not an outlier, but now in mass, it's definitely out there. Mean value, it's way out here. So if we want to take a look at that, I can't remember what a Fahrenheit is because let's say I'm, not a doctor. I can click on, some other object that's kind of in the mean or the middle of the distribution, and I can remind myself what a Fahrenheit is. And so give it a second, and it'll load up, and then it also selects that Fahrenheit here. So I can look at that and go, oh, okay. The Fahrenheit is this part of the anatomy. And I can remind myself what a normal Fahrenheit looks like in this case. And so now let's go back where we were. And now let's go back to our outlier here. So I click on the outlier over here. Now I can load this one. So now that I've reminded myself what normal looks like, I can now load the outlier and take a look at what's going on. And okay, maybe it's not obvious what's happening. I look at my list of objects up here. So if I select through my objects, I can see for whatever reason, this particular scan, in this case, has the nerve here selected on the left side, has the nerve selected, segmented on the right-hand side. And then what is labeled as the pharynx is actually part of the mandible combined with the pharynx. And so that helps explain why it's an outlier. This is also not something I created. This is an actual error in the data. So in this case, this particular segmentation is definitely screwed up. So this is the kind of thing that we want to find to avoid errors if we're going to train machine learning algorithms on this kind of data later. This kind of problem can snowball into bigger problems. So if you're trying to train something to detect or delineate pharyngexes from cone beam CT scans, this one scan with this additional jawbone or mandible attached here as part of that can definitely introduce significant error that's going to result in a poor algorithm. So in this case, to fix this, what I would actually do, I'd select that object. There's two separate pieces, so I can actually easily click on the split capability here. And then when I hit split, what it's going to do is going to find all of the objects that are not touching, or all the voxels that are not touching one another, and then separate them out. So in this case, I got a bunch of little single voxels, which are sort of peppered through. You can see here something going on there. So what I'm going to do is I'm just going to take all those and just get rid of them. I'm going to throw those out. And then the way it Okay, now I've got two objects here that are split. The labeling's incorrect. So I'm gonna actually label this one as the pharynx. So I can go down here, choose my anatomy. I can change to pharynx. And now that one's labeled properly. And then I can go to the lower jaw and I can fix that one. So this is no longer the pharynx. This is the jawbone, and I need the lower, I can't remember which one of these it is. This one, so region lower. So now I've got that labeled as lower jawbone. Sorry about that. Keep on getting spam phone calls. And then I'm going to save that. Now that's saved. I can go back to, I'm going to close that now. I'm going to go back to our quality assurance tab, and this is going to get updated. So now that's been updated, and our object's been moved down here. Now that it's been fixed. And now we can go check our list again and see what the next biggest outlier is. And, you can kind of just, repeat that process to continually find errors and that kind of thing. Let's see. Another important thing to be able to do is exporting. So let me find that folder again. So I'm going to take this, and then I'm going to go to the export, and I'm going to put that data here. So this finds all the proseg files and all the objects that I have, and then if I want to do an export, let me find a folder. Make another one here. And I'm going to export into this folder, and then it's going to tell me the files that it would create. So I can create a few different output formats here. VTK, Nifty, a couple others. If you need something else, you can always let us know, and we can add it generally. In this case, I'm not going to take the time to go through and export all of these, but I will export the metadata. So, and show you what that is. So, if I hit this export all, it would now go through each of these data files and create the segmentation files from those. When I click the export metadata, what it does is it goes through each of these, and then it's going to create a CSV file that you can load into Microsoft Excel, for example, or Python or whatever you want, and it'll give you various metrics about all of the different objects that you have. So like volume, the attributes that have been assigned to them, which scans they're in, dimensions, that kind of thing. So there, we're done now. So now it's created these three files. So let's take a look at this one. And in the scans file, it tells us, the folder that it came from, the file that it came from, width, height, and depth of the three-dimensional volume, the spacing, X, Y, and Z spacing, origin, units, and in this case, operator time is all 0 because we imported this from some other software tools. So this shows that there's 480 scans that have been as part of this data set, and tracks that information there. This file here, instances, so in the scan CSV, you have one row per scan, and in this case, you have however many objects there are. So if we look at scan ID number 1, we have 32 objects. And then these are the different object identifiers here. And then these are the different values that are used in the segmentation for that object. And so scan ID here correlates to this value over here. So, the scan number over here. So you can connect the dots between the different files. And then if we look at one particular object, so let's say, object number 4 and scan number 1, we can find the center XYZ position of it. We have the volume of it. We have the mass. We have the intensity value. The mass is, I think, actually off by possibly 1,000 in this case because the intensity value is negative. And then you have the min-max mean, median, standard deviation of the intensity values here. And then these are all of the attributes that were assigned to each of those objects. And so in this case, you have a column for each attribute category, and then you have the value that was set. So in this case, jawbone, and then in this case, the region is either upper or lower. For example, blanket indicates that there was no value set for that particular attribute. So you have all of this information here, which can then be imported into an algorithm training process if you're going to be training your algorithm to do something, to find an object or something like that. So you need access to that kind of metadata. And then lastly, this operator file. This is more. It's not really going to be that useful, but it tells you different information about the steps that an operator went through for each of the scans, which if the operator tab was working, and I could show you, you could see that information. Give me a second to try one more time.
David, about 4 minutes ago, we had a question about, can it generate mesh files? Oh, yeah. So I'm assuming you mean the isosurface. So from an isosurface perspective, is it from? Can the person that asked the question clarify from the segmentation, or is it from like the bone surface, for example, like this? So I'm gonna, while we're figuring that out. So if I want to select a surface from bone like this one, I can choose that surface there. And then you can hit extract surface right here, and then that will generate like a PLY file or a STL file. And so let's put that in here. Test.ply. And then it pulls it out. And then you can load that file in some other software, which for fun, I'll show you one of our other software tools. So there's another one of our software tools here, Stratovan Checkpoint. And so find the folder, I just drop that into. So here's the PLY file there. And I can here. Now I have the surface here. For fun, just to give you a little bit of information about this particular software. This software is meant for placing three-dimensional landmark points and measuring morphology of shapes, three-dimensional shapes. So I've got more context of the question, David, if you're ready for it. They're saying, yes, I meant a PLY, OBJ, or STL file. Can it also generate those from a region of interest you've defined?
I don't think currently, but that is something we can add. Yeah, there's a way we can do that. So okay, so let's say I want to do it for the jawbone here, the mandible. So I'm going to select the mandible here, lower mandible, which is that green region there. And then what I can do is I can export just that region. So I'm going to do isolate selected object and hit apply. And then what that's going to do is, let me put it in a folder. A second. Make yet another folder here. Put it in that folder, and I'll give it mandible. Okay, and then I'm going to save that. Isolated region saved. Okay, so now that's been exported. Into here. I'm going to go back, and now I'm going to load this. Here. And now it's just the mandible that's there. So all the other stuff has been removed. So just the mandible. Even like the roots of the teeth and everything have been removed from that based on the segmentation. And if I go to the surface tab, I can now choose a surface. And you can look here. So the red, if you look here, there's a red showing where the surface is being pulled out.